As you remember from the beginning of the semester, we talked about how viruses are non-living. Viruses are also very, very small. We cannot see them with the microscopes that we have in this classroom. They are very tiny. So the pictures at the bottom of the screen are ones that have been magnified tremendously. So viruses are not living, basically because they do not meet the criteria of life. Remember, there was certain things that all living things have. First, all living things have to be made of cells. Viruses are not made of cells, known as acellular, so they can't be alive. They do not have a true metabolism. In other words, they can't take in stuff and turn it into energy. Just like we take in oxygen, we take in carbohydrates, we make energy. Viruses cannot do that. Viruses rely on who's ever the host, whether it's us, a plant, another mammal, for their energy and the enzymes. Enzymes, remember, get things done in the body. So viruses rely on other organisms for their energy and for their enzymes. Technically, viruses don't grow. They do reproduce, though. They are completely parasitic, so they must enter a host cell to replicate. A lot of people say, well, they seem like they're living. The reason they seem like they're living is because they replicate and they do have either DNA or RNA. These are the main parts to what a virus has. There is a protein coat, also known as the capsid, and that capsid is going to determine the shape of the virus. It could be helical, polyhedral, complex. There's a few other shapes. It will have a nucleic acid core, whether that is DNA or RNA. It usually has some type of tail sheath and some tail fibers, and it's those fibers that are able to embed itself into that host cell. Bacteriophage, we're going to hear this term throughout this whole chapter. Bacteriophage, it's a virus that actually attacks a bacteria. So just like viruses attack animals, just like viruses attack plants, viruses can attack bacteria. To us, bacteria seem very small, but to a virus, bacteria is very large. So remember, viruses are extremely tiny. And if you look at the screen, that bacteriophage that says bacteriophage T4, that is a real picture of that virus. And then off to the right is the computer-generated picture. So off to the right, you can see that there's a head with the DNA inside. That's that capsule. It shows you that tail sheath. It shows you the tail fibers. And again, here's just another picture of a virus. Nucleic acid is surrounded by the capsid, or the protein coat. Larger viruses will be surrounded by an envelope. It just gives them an extra protection. And it's the nucleic acid, that DNA or RNA, contains the instructions for making more of that virus. So if you look at the picture, you have the envelope, but you still have the capsid and you still have the DNA. And the little tail fibers are those little projections coming out of the envelope. The virus finds and attaches to a specific host cell. Not all viruses can just go out and attack every cell out there. They are very host specific. Only certain viruses can attack us. Only certain viruses can attack certain plants. Like there's the tobacco virus. It can only attack tobacco plants. What happens is the virus comes along and the little tail fibers can attach to the membrane of the cell of the organism. And once it's attached, then the DNA can get inside the cell and start replicating and make more of the virus. So the attachment process resembles kind of like a spaceship docking procedure. I'm going to have some kind of animated video of that a little bit later on. And the picture off to the right there is a virus attached to a cell. The blue thing is the virus attached to the cell. So again, viruses are host specific. The attachment site and the receptor sites need to fit together like a lock and a key. So the attachment site on the cell and the receptor site on the virus have to fit together. If they don't fit, the virus is not going to be able to attack that cell. So we could inhale many viruses, but they're not necessarily going to attack us, only if the attachment site and the receptor site can fit together like a lock and a key. So the attachment site of the virus and the receptor site on the host have to fit together. There's some basic steps that go to how a virus reproduces and how it attacks cells. Here are the basic steps. There's attachment, penetration, biosynthesis, maturation, and release. These are the 5 main things that have to happen in order for a cell to be able to attack and get inside of the cell and start reproducing. The first basic step is that attachment process. The attachment site on the tail fibers of the virus combined with the receptor sites on the cell. If that can't happen, the virus can't attack anything. Once that has taken place, once the virus has attached, then the DNA and RNA can get inside the host cell. That's known as penetration. Once the DNA and RNA is inside the host cell, it will make many copies of its capsid and of the DNA, so it's making many copies of itself. That's known as biosynthesis. Remember, bio means living, synthesis means to make. So here, the bio part is that DNA, making many, many copies of that DNA or RNA, and then it also makes many copies of the capsid. The capsids and DNA come together then. And then you have the maturation. The DNA, RNA, and the capsids are assembled to produce thousands of viral particles. And the last part is the release. The new virus leaves the cell, and usually the cell will die. So you had one virus enter the cell, or one virus attached to the cell. The DNA gets penetrated. The DNA and capsids are reproduced over and over again. The DNA and capsids are put together, and then you have thousands of viruses being released. And if you remind me, I can show you a couple videos kind of demonstrating this. Each virus can only attach to one kind of cell due to that lock and key type of fit. So that's just showing you here that you have those receptor sites and attachment site of the virus. The red part being the attachment site and the bluish part being the receptor sites. There are 2 ways a virus can actually get inside of the cell. Either the virus is attached on the outside and it just puts the DNA or RNA into the cell. So it says virus injects nucleic acid into the cell and leaves the capsid on the outside. That's one way, as the picture is demonstrating. And this would kind of be demonstrating the penetration part. It already attached, now it's putting the DNA inside. The other way that a virus can get in is through endocytosis. Endocytosis is when a cell engulfs large particles. We briefly learned that towards the beginning of the semester. Endocytosis. So a cell will engulf the whole virus. So the entire virus enters the cell, and then it gets wrapped in a little membrane, as you can see in the diagram. Once the DNA is in the cell, from there, depending on what type of virus it is, it's going to depend on what's going to happen. There are 2 viral cycles, the lytic cycle and the lysogenic cycle. With the lytic cycle, the virus attaches to the cell. Virus penetrates cell membrane and injects the nucleic acid. Viral nucleic acid replicates using host cellular machinery. New viral acids are packaged into viral particles and released from the cell. The host may be destroyed in the process. So that's kind of those steps: attachment, penetration, biosynthesis, maturation, release. They just combined one of them, so there's only 4 steps here. So the lytic cycle is those basic 5 steps that we've been talking about. The virus uses the cell's material and energy to replicate. The new viruses burst from the cell is killed, new viruses are released into the body to infect and kill more cells. Once your immune system starts working, then your immune system hopefully will take over and get rid of the viruses. But until then, you are sick. Okay, so the lytic cycle, we already have attachment, and then we have the penetration. The viral DNA is replicated. So we have biosynthesis and maturation together. So this is showing biosynthesis and maturation. So right there you have the biosynthesis, and then you have maturation, and then you have the release. And all those viruses now can go and attack more of your cells until your immune system starts catching up with it. Because at first, viruses aren't detected by your immune system. That's why you get sick. Okay, so attachment, penetration, biosynthesis, maturation, and release. The lysogenic cycle is a little bit more complicated, and you'll have another handout that talks about both of these cycles in more detail. But with the lysogenic cycle, the viral DNA is mixed into the host DNA and is now called a provirus. The provirus DNA is copied every time that cell replicates. Viruses remain dormant until a trigger will activate them. So for a while you have this virus in your body, and it's every time that cell replicates, so does the virus. So if the cell replicates 1,000 times, that virus has been replicated 1,000 times, and maybe that virus came in and it's 10 years later before the trigger happens. Once that trigger occurs, then all of those viruses all at once start attacking your cell. So all viruses enter a lytic cycle simultaneously. All the host cells are killed at once. Again, it'll enter. And this is what's different, and it becomes part of the host DNA. Eventually, some trigger causes the virus to enter a lytic phase, releasing them and killing the host cell. Like I said, we'll talk more about this using another handout. Example of lysogenic viruses are cold sores, herpes, hepatitis B, HIV, chickenpox. So think about when you have chickenpox, it goes away, but that virus never really leaves your body. When you have a virus, that virus is in your body forever. Your body never really gets rid of it. So with chickenpox later on, you can get something called shingles. HIV. People oftentimes get HIV in the teens and early 20s, but they don't show any symptoms until their late 20s and 30s because of that delay. HIV is a lysogenic provirus that affects the white blood cells. Viruses enter and are replicated by cells. Eventually, all the white blood cells are infected. When triggered, proviruses will kill all the white blood cells at once, and that's when the person has AIDS. A retrovirus. Retroviruses have RNA and contain special enzymes called reverse transcriptase to change the RNA into DNA. Remember, DNA is usually changed into RNA. Okay, viral drugs. Rarely has it been possible to find a drug that interferes with a viral reproduction without interfering with your or the host metabolism. Since viruses use the host cell for reproduction, it is very difficult to find drugs that will destroy the virus without damaging or destroying the host cells. In other words, if the virus is in you, it's in your cells. It's hard to get rid of that virus without destroying your cells. If we're destroying your cells, we're killing you. In other words, it's hard to find a medicine that affects the virus but does not affect your cells.